Next, 19th question here. So in image, colored and full endosperm is dominant over the colorless and shrunken endosperm. Means we have to choose this colorless and shrunken as a recessive. In F1 generation was subjected to test class. So here giving colored C and full endosperm F. These two are dominant and we have to take one parent, dominant parent, and crossing with recessive parent. Here recessive parents are colorless small c and shrunken endosperm small f. So when we cross between these dominant and recessive parents, and in F1 generation we will get, generally what we get in F1 generation means, in which we, according to Mendel, we will get dominant character only, means we will get colored and full endosperm only, full endosperm. So when this F1 generation is subjected to test cross. So here test cross means crossing of F1 with recessive parents is called test cross. When it's F1 subjected to test cross, then there are 4 phenotypes will be produced. Here giving in produce 4 phenotypic in the following percentage. So what are the phenotypic here producing? First one here colored full maize produce how much? 48%. The second one colored shrunken here colored full and shrunken endosperm are present. How much? Shrunken endosperm 5%. And the third one colorless but full endosperms are how much? Here 7%. And the fourth one colorless shrunken. So this was produced 40%. So these are the given data. So what we have to do, we have to find out the distance between genes, between two non-allelic genes. So these questions directly from we have to find out the distance between genes. So how we can find out, how can we measure? By using genetic map. So before using genetic map to find out the distance between genes, first let us know in which what are the parental combination and what are the recombinant combination. So we know that here, so simply we can see here dominant parent we have color and full endosperm. Recessive parents here, recessive parents we have colorless and shrunken. First we have to find out, so we have to segregate which is parental character and which is dominant, which is recombinant combinants. So for example here colorless, so here colored and full endosperms are dominant character. So how much? 48%. Okay, plus, and here colored and shrunken, and these two are what? Generally recombinant character. Why? Because in these two characters, new characters will come in this generation. New characters will come, so it is recombinant character. Here colorless means this recessive character, and here full endosperm and dominant character. So this also recombinant character. And here see colorless and shrunken. Here colorless and shrunken, these two are what? Recessive character. So how much recessive character here? 40%. So 48% and 40%, both are parental characters. Both are parental characters. So here 40 means there are 88% of characters are parental characters. How much? 88 parental characters are there in this 4 progeny. And the rest of these two, like here colored shrunken 5%, and here colorless full endosperms 7%. So total 12%. So 12% is what? Recombinant character. Remember, 12% is recombinant character, and here 88% is parental characters. Okay, so here we have to find out genetic map. So remember, genetic map is always to find out the distance between here. So how to find out means in genetic map, so the distance between, here I am writing, distance between genes is exactly equal to the percentage of recombinant. Very important. Exactly equal to the percentage of recombinant. Okay, for example, how much percentage of recombinants here we have? 12% of recombinant we have, and is exactly equal to the distance between genes. So 12 units, genetic 12 units is exactly equal to this 12% of recombinants. So like that. So these 12 units are also we can use centimorgan units or genetic map unit. Also we can use map unit. So here remember, so here the distance between gene, remember this important, distance between gene always is exactly equal to the distance of the gene. Sorry, distance of the or the percentage of the recombinants. So how much recombinants? Here we have 12%. So this 12% recombinants is exactly equal to the distance between genes. So 12% means 12 units are the distance between two genes here we have. So here the right answer for this question is 12 units. So here where we have 12 units here. So right answer is fourth one, 12 units. Okay, clear now? Right. Here next question is 20. So here choose the correct statement from the following. Here Clistogamous flower always exhibit autogamy. Chasmogamous flower always exhibit geitonogamy. Okay, Clistogamous flower exhibit both autogamy, geitonogamy, and Chasmogamous flower never exhibit autogamy. So here we have four. So these questions which is came from types of flowers based on their opening and closing. Here first we will discuss Clisto flower. So Clistogamous flower, they never. So what are the Clistogamous flower never open? They never open because the reproductive organs are never exposed, because the reproductive organs are never exposed to the pollen, outside pollen. Hence they perform only self pollination. Here self pollination is nothing but autogamy. So it is quite opposite to the Clistogamous flower. Sorry, Chasmo flower. So here Chasmogamous flower means the flower always open because they want to, the reproductive organs are exposed to the foreign pollen grain also. Your reproductive organs are nothing but they are ovary, like a pistil and stamens. Reproductive organs are always exposed to the foreign pollen. So hence there is a chance of cross, or also called gaetano. So not only that, but also there is a sometime this Chasmogamous flower also exhibit autogamy. Both Chasmogamous flower, autogamy means self. So here Chasmogamous flower all both, it exhibit both autogamy and geitonogamy, means self pollination and cross pollination. But remember, Clistogamous flower never open, due to which they never expose their reproductive organs to the any foreign pollen grain. Hence they only has the they only perform self pollination, that is autogamy. So here see this Clistogamous flower always exhibit autogamy. So this is the right one. Why? Because Chasmogamous flower always exhibit, no, not exhibit geitonogamy. Always exhibit geitonogamy means wrong. Why? Because sometimes the Chasmogamous flower also perform autogamy. So hence this statement is wrong. Clistogamous flower exhibit both? No. Why? Because Clistogamous flower exhibit only autogamy, not geitonogamy. So Chasmogamous flower never exhibit autogamy? No. It can be exhibit autogamy and both. Chasmogamous flower exhibit both autogamy and geitonogamy. So the right answer is first one. So here 21st question, identify the wrong disease symptoms. Here we have to identify wrong disease symptoms with example. Disease symptoms, here we have to identify wrong disease statement. So mosaic, yes, these symptoms cause tobacco mosaic disease. This is a right statement. Wind clearing, yes, bendy wind clearing. And here breaking of flowers, so tulip mosaic break. So malformation is not matching with swollen shoot of potato. Hence here we here asking wrong disease symptoms. So here right answer. Why this right answer we will discuss. So generally mosaic. So what are the symptoms of general mosaic? Here mosaic symptoms. Mosaic symptom. So mosaic is a generally the symptoms from viral infection in plants. So here what are the characteristic symptoms of mosaic means? Here irregular patterns of alternative light green and like yellow patterns and dark green patterns are seen on leaf. Dark green pattern seen on leaves. Irregular patterns are generally seen with this color. So that is the characteristic feature of mosaic. Mosaic symptoms which mainly due to caused by virus called tobacco mosaic virus, and this leads to cause tobacco mosaic disease. Okay, hence this statement is right generally. So but here we have to choose only wrong disease symptoms. Now coming to wind clearing. So here wind clearing means loss of green color in the veins region of leaf. So we know that here like we have leaves, so these all are veins. Okay, so at this veins region there is a loss of green color. So once there is a loss of green color in the veins of leaf, so which making them appear un yellow white in color. So hence due to loss of green color and there is a like formation of yellow color. So yellow whitish color structure will form in why because of there is a loss of green color. So what happened here in vein clearing means, so reason is to break down chlorophyll pigment. So generally chlorophyll pigment is responsible for green color, but what happened here, chlorophyll pigment get breakdown. So due to breakdown of this chlorophyll pigment within the veins and veinlets, so there is a formation of yellowish or whitish structure. Such symptoms are called vein clearing, which is generally seen in bendy. Okay, now coming to malformation. Here I'm writing malformation. So this malformation it is due to uncontrolled cell division. Very important. Malformation occur generally due to uncontrolled cell division. Do you know this uncontrolled cell division? Also we can use another technical terms, that is hyper. So due to uncontrolled cell division, it leads to cause malformation symptoms. And not only that, it is also due to abnormal cell enlargement. Generally here cell become within must and should be enlarged within its shape and structures. But here what happened? Abnormally cell enlargement will occur. Enlargement. Abnormally cell become enlarged. So this technical term here we use hypertrophy. Here hypertrophy means abnormally cell enlargement. Hyperplasia means uncontrolled cell division. So this will occur, and due to occur these two, uncontrolled cell division and abnormally cell enlargement, which leads to swollen tissue. Okay, swollen tissue of in which plant means especially coca plants. But here in option giving potato plant, so in potato does not occur. Generally potato it store food, hence there appear like a swollen part. Okay, why potato appear like a swollen part means because of it store food in its stem. Okay, but not due to this uncontrolled cell division of abnormally cell enlargement. But this type of enlargement are seen only in coca plant. Here very important, coca plant only we have seen this abnormally cell division, uncontrolled cell division, abnormally cell enlargement. So that's why this question is wrong. Okay, so breaking of flowers generally which is seen in tulip mosaic break. So here right answer is third one. Yeah, next one, 22nd question here, polyembryony in citrus is due to. So we'll discuss what exactly poly. So this polyembryony concept generally which is present in single seed only, remember. So here polyembryony form without fertilization or sometime with fertilization. So here what exactly mean of this poly? So in name only it is giving poly means many, embryo means embryo. So many embryos in a single seed. Generally single seed has only one embryo, one seed only. But in this polyembryonic condition, there are many seeds in many embryos in a single seed. Such situation is called polyembryony. So we can define in simple term, development of, here how we define means development of more than one embryo in a single seed is called polyembryony. More than one embryo in a single seed is called polyembryony. Generally these polyembryony conditions mostly seen in gymnosperm plant. So most commonly found in gymnosperm plants. Very important. Most commonly found in gymnosperm plant. Only few are angiosperm. Only few are seen in angiosperm plant. Few embryos are seen in angiosperm plant, but many seen in gymnosperm plant. Remember. And here, and this polyembryony concept was given by a scientist, Anton van Leeuwenhoek. Very important. Anton van Leeuwenhoek proposed this concept in citrus. So here generally these polyembryony conditions are developed from where we have different types of polyembryony, but here we need to discuss as per NCERT. Here in what happened in polyembryony, this embryo developed from new cellus. Very important, new cellus, and also integuments of ovules. So for example where we have see, so generally see this is what placenta. So stalk of the ovule is called funicule, and here hilum, and this is our body. So here body of ovule, and here we have hilum. This is what funicule, and this is placenta. From placenta only the ovule is, and this body of ovule contain two important structure. So inside which we have embryo sac, also called female gametophyte, and around which we have two protective layer around embryo sac. So that is like this 1 and 2. So these two are called integuments. So this integument here, this integument which protect this, there are various tissues which is present inside the integuments, okay, around the embryo sac. These tissues are called nucellus cells. So here what happened, so this development of polyembryony occur either from nucellus or from integuments, either nucellus and integuments. How this generally which occur means, here very important, here nucellus cells, division of nucellus cells, here there is a division of nucellus cells which occur, division of nucellus cells or tissues. So when there is a division of nucellus cells which occur, and these nucellus cells, some of the nucellus cells which protrude into the embryo sac. How means I will show you, protrude into embryo sac. For example, see, this is embryo sac. So here we have various nucellus cells. Just think, in among some, there are number of nucellus cells are present, among which some cells are undergoes rapid division. Then the divide nucellus cells which is protrude into the embryo sacs like this, protrude into the embryo sacs. So this protrude nucellus structure later modified into embryo. So if how many nucellus cells which is protrude inside the embryo sac, and each and every protrude nucellus cell later develop into embryo. So hence there are various means polyembryo conditions are seen. Polyembryo condition are seen. Okay, so very important, polyembryo conditions are generally present or due to division of nucellus cells which is protrude inside the embryo sac, and later these protrude nucellus cells later develop into many embryos. So like that, but here the true embryos comes by in zygote only. True embryo, generally here we have true embryo. For example, here I will show you, just wait here. Yeah, here for example, here we have embryo sac. So here we have egg. Okay, so first primary male gamete fuse with egg cells, then it result to formation of zygote. Here zygote will develop, and this zygote later develop into embryo. The developed embryo is called true embryo. Why? Because the embryo which is developed from the zygote, such embryos are called true embryo. So here true embryo will be developed by fertilization. But here what happened, so due to various divisions of nucellus cells, and those divide nucellus cells which is protrude inside the what embryo sacs, and these protrude are infiltrated nucellus cells into the embryo sacs, later develop into embryo. These embryos are false embryo. Why? Because this is not undergoes any fertilization to develop embryo, and these false embryos having many, there are many false embryos are protrude inside these embryo sacs, and this is known as the poly. So here polyembryony form without fertilization. Remember, very important, polyembryony conditions form without fertilization. Okay, remember. So here, so by this analysis we can analyze which is the right one here. Just wait, sorry. So here if we see the options here, so here, yeah, so here there are 4 options are there now. Yeah, so here division of nucellular cells, very important, division of nucellular cell, and it is penetration into embryo. Penetrate means protrude. Okay, protrude or penetration we can use. So into the embryo, then it leads to be this. So right answer is this one. And why this wrong answer means, here see, development of perisperm cells, no. So does not, perisperm cell does not involve in formation of any kind of embryo covered, hence this statement is wrong. Fertilization of antipodal cells inside embryo cells, generally antipodal cell does not involve in any fertilization. Okay, and next, division of synergid cells inside the embryo sacs. So this statement is also wrong. So right answer is first one. Okay, so next 23rd question here. So here the sequence of nucleotides of mRNA, here giving sequence of nucleotide on mRNA and the sequence of respective amino acids giving polypeptide chain below. So what we have to do, we have to find out nucleotide sequence of DNA template strand from which this mRNA was transcribed. So we know that, first let's start discussion on these questions. Yeah, generally we know that there is a central dogma of, so in which what happens, central dogma DNA has genetic information, and that information transcribed in the form of mRNA. See, this is mRNA by a process called translation process, and this mRNA information translate into protein, and by process called translation process. So this is transcription, this is translation. By these two process, the information which is present in DNA, which is in the form of protein only form. Okay, finally. So here generally we know that the DNA has double stranded 5 prime to 3 prime and 3 prime to 5 prime. So among these two strand, one of the strand act as a template strand. Very important. Mostly template strand here 3 prime to 5 prime. Why? Because always RNA synthesis here. mRNA synthesis always occur in 5 prime to 3 prime direction. Very important. So hence in a double strand of DNA, this 3 prime to 5 prime is only act as a template strand. Okay, so here already in questions there is a giving of mRNA sequence. See, so how mRNA sequence is giving TAC and sorry, here mRNA sequence here already here given now. So sorry, just yeah. So here mRNA sequence here giving mRNA sequence, this mRNA 5 prime to 3 prime, like here I'm clearly writing 5 prime to 3 prime. So, yeah. So here AUG sequence is code for methionine amino acids, and phenylalanine. AUG again it code for methionine, and CCU this one, and here GUU, and here U, sorry, just what is this? Yeah, U double A here again 3 prime U double A. So this is what giving nucleotide sequence on mRNA. So we have to find out which strand of DNA it has. Okay, right. So generally here you need to remember two important concepts. So generally in DNA we have 4 nucleotides. Even in RNA also we have 4 nucleotides. The major differences between DNA and RNA nucleotide is in DNA we have thymidine, but in RNA we have uridine or uracil. We can simply use uracil. So thymidine present in DNA, and RNA we have uracil or uridine. Okay, and one more important note here. So remember, as per Chargaff rule, so here purines always pair with pyrimidines only. Purines pair with pyrimidine. Here purines are like adenine and guanine. Here pyrimidines are cytosine, and in case DNA that is thymidine, and in case RNA that is uracil. So here remember, adenine always pair with thymidine in DNA, or for example in RNA means A pair with uracil. Remember, G always pair with cytosine only, always pair with cytosine. But adenine in case of DNA it is paired with T thymidine. In case of RNA, adenine pair with uracil. So that is a major difference. So if you know this concept, then we can easily identify which is the DNA sequence of template strand it has. Okay, right. See, so 5 prime to 3 prime means definitely the template strand must and should be has in this sequence, like 3 prime to 5 prime sequence. So here identify A. So here A. So generally these nucleotide sequence are present on mRNA. So now we have to focus on DNA sequence. DNA. So A pair with T. So here we have A pair with T, and here U generally RNA, but here U pair with not U. Instead of U, what we have? A, G, always C. Okay, na? Here G always C. And coming to this 3 nucleotide sequence, triple U means here triple A, and here AUG same T A C, and double C U. C means G, U means A, and G U. Here G means C, U W means A, and U here what? U double A. U means A, double A means double T. So this is the sequence of template DNA strand. So we have to choose where we have this TAC here, TAC triple A, and then TAC here, and DA here, C double A here, and A double T. So the right answer is this one. Okay, like that we have to find out the DNA template sequence once if we giving mRNA sequence. In sometime there is a giving a template DNA strand, then we have to find out mRNA. Then we use this U is play important role. Why? Because we have to find out, and it include U only. But here already given mRNA sequence, then we have to find out only DNA strands. So this is the first MCQ is the right answer for this 23rd questions. Yeah, next one, 24th question here. Family Fabaceae differ from Solanaceae and Liliaceae in relation to stamen. Here, stamen relation. Identify the characteristics specific to family Fabaceae. We have to identify the family, and that family must and should be differ from Solanaceae and Liliaceae. So, and this question is completely from in relation to stamen only. Stamen. Do you know stamen is the male reproductive organ. This stamen has two important part like this. Okay, so this part is called filaments and this part is called anther plus filaments together form a single stamen. Remember, the group of stamens together form a structure called androecium and it is indicated with EA. Okay, fine. So generally these stamens are some stamens here I am writing just wait. Here stamens may be free or may be fused, may be free and fused. If free stamens are called poly and fused stamens are called gamoandrous. Gamoandrous fused stamens and polyandrous free stamen. And generally in polyandrous we have two different types like tetra stamen and here didynamous stamen. So didynamous stamen in which four stamens are there. In tetradynamous we have six stamens. Remember. So among six, two are outside, four are inside. Out, two are inside. And didynamous equally two outside, two inside. Out and two inside. Like that we have. Do you know this tetradynamous is a characteristic feature of Brassicaceae family members, which is a Brassicaceae. In Brassicaceae family, like Brassicaceae means Cruciferaceae family. Brassicaceae are Cruciferaceae family characteristic is tetra, and didynamous is a characteristic feature of Labiatae or Lamiaceae family or Lamiaceae family, like Asimum sanctum. Okay. Now coming to here, fused gamo, and these gamoandrous in which again we have two types. Their cohesion of stamen or adhesion of stamen. Here I am writing here. This gamoandrous again we have two types based on here the fusion of stamens with same stamen or any other part. They are based on that we have cohesion of stamen and adhesion of stamen. Here cohesion of stamen means stamens only together form like a bundles. Here adhesion means the stamen which is attached to the other than stamen, like sepals, petals or any other structures. In cohesion of stamen, again we have three different types. Very important. In cohesion of stamen, we have three different types. They are adelphous, and second one is syngenesis, and the third one is syn. So here adelphous means simply in adelphi only filaments are fused. Means anther free and only filaments are fused. How means like this. For example, here we have a group of stamens like this in which filaments are only fused like this. Filaments are only fused but their anther is free. Here anther free but filaments are fused. Such condition is called adelphous. So we have generally there are three types of adelphous: monoadelphous, diadelphous, polyadelphous. In monoadelphous is the most characteristic feature of Malvaceae family. Is the most characteristic feature of Malvaceae family. So in Malvaceae like China rose, like hibiscus, rosa, sebensis, cotton. Okay. So in which we have only monoadelphous, and these monoadelphous having mono very important monothecus. Okay, mono anther it is there in monoadelphous. Now coming to diadelphous. So diadelphous means in which the stamens, the group of stamens are arranged in two bundles like this, two bundles in which filaments are fused but their anther is free. Two bundles means 9 plus 1. Here 9 bundles are generally close to each other and one is separate like this. And these diadelphous conditions are seen very important in family called Fabaceae family. Very important. Fabaceae family. So in Fabaceae family we have seen diadelphous condition in which the group of stamens are formed into two bundles in which one bundles contain 9 stamens and one bundle contain 1 stamen, like that seen in Fabaceae plant. And this diadelphous condition of diadelphous of stamens are generally having dithecous anther. Very important. They have dithecous anther, bilobed anther, bilobe or dithecous anther. And polyadelphous conditions are generally seen in citrus plant, in which there are group of stamens are formed into group of bundles. So many bundles are formed by group of stamens. Okay, seen in citrus. Like that we have different types of adelphous. So what is syngenesis? Here it is quite opposite to the adelphous. So how means in which anther is fused but filaments are free. Very important. So these syngenesis conditions are generally seen in like Asteraceae family. Very important here. Syngenesis. In syngenesis, how syngenesis is formed means here there is a group of filaments are free but their anther is fused like this. So here anther fused but filaments are free, and this type of cohesion is called syngenesis. And syngenesis is the characteristic features which is seen in composite family. Also we can use another term for composite family, that is Asteraceae family. So remember one important note here. So composite family or Asteraceae family is the largest family, very important. It is the largest family among 202. Among two, we have 202. As per Bentham and Hooker classification, we have 202 families. Among 202 family, the largest family is composite or Asteraceae family, and it has syngenesis type of cohesion. Okay, right. Now coming to synandrous. In synandrous, here, just wait. Yeah, synandrous. Syn means fusion. Androus here, synandrous means in which both means anther and filaments both are fused. Anther and filaments both are fused only. How means like this? See, so here filaments fused and even anther also fused. So anther fused and filaments also fused. So this type of condition is called synandrous. This type of synandrous generally which is seen in a family called Cucurbitaceae family. Very important. So in Cucurbitaceae family, we have seen synandrous means in which both filaments and anthers are fused together, and the syngenesis are seen in Asteraceae family or composite family, in which only anther fused, filaments are free, and in Adelphus conditions, in which what anther free, filament is only fused. And this Adelphus, there are three types. We have monoadelphus, diadelphus, and polyadelphus. In monoadelphus, mostly common in the group of stamen form into a single bundle, hence it is monoadelphus, are commonly seen in family called Malvaceae. In diadelphus are seen in Fabaceae, in which the group of stamens form into two bundles. And in coming to polyadelphus, in the group of stamens form into many bundles, which is seen in citrus. So like that we have different types of cohesion of stamens. So within here, and there are various type of adhesion of stamens we will discuss. Just wait. Adelphus of stamen. Yes, adhesion of stamens. Adhesion of stamen. So here adhesion of stamen means the stamen attached to the other floral part, other floral parts, except stamens together form cohesion, and stamen attached to the any other parts of the plants called adhesion. So in which we have different types of adhesions like epicephalous, in which what happens, stamens attached to the sepals, stamen attached to the sepals. And one more condition we have, epipetalous. It is most common and very important, epipetalous, in which stamen attached to the petals. Generally this stamen attached to the petals are generally seen in very important family, that is Solanaceae family. In Solanaceae family, we have seen epipetalous conditions. And also in sometime there is a epiphyllous stamen. It means the stamen which is attached to the phyllis. Here phyllis means perianth. Generally perianth means in which there is no sepals and petals both are integrated to each other, form a single structure, perianth. So if the stamen attached to the perianth, such stamen is called epiphyllous stamen, which is the most common in Liliaceae family. Very important. In Liliaceae family, we have seen epiphyllous stamen. In Solanaceae family, we have seen epipetalous conditions. Okay. And in Fabaceae family, we have seen diadelphous stamen. Remember. Okay. Like that, we have different types. So here we have to analyze now these questions here. So what here exactly asking? So by this analysis, we can conclude in Fabaceae, which is differ from Solanaceae and Liliaceae. So here, how they are differ with respect to their stamen. And remember one more important point. Generally, in Fabaceae has 10 stamen. Remember, 10 stamen, and each stamen has like diathecus. Very important. They are diathecus. Each lobe has 2 theca. Okay, but it is a bilobed means each lobe contains 2 theca, hence diathecus. If bilobed means completely there are 4 thecas are present, tetrathecus. Okay, remember each lobe, here we need to consider each lobe consists of how many theca, means 2 theca, hence also we can use bilobe. Stamens are seen in Fabaceae 10 stamen, and these 10 stamens which are arranged into 2 group, 9 plus 1, and this 9 2 group means diadelphous conditions, in which 9 stamens are together and 1 stamen is completely separate. 9 plus 2 arrangement. And coming to Solanaceae, in Solanaceae there are 5 stamens are there. Very important. 5 stamen. In Fabaceae 10 stamen. Solanaceae, in Solanaceae only 5 stamens. Remember. And very important, in which epipetalous conditions are seen. Epipetalous. Okay. And here also the stamens, the anther is diathecous anther, means they are also having bilobed anther. We have to differentiate. Okay. Now coming to Liliaceae. In Liliaceae there are 6 stamen. Very important. there are how many stamens? 6 stamens in Liliaceae, and these 6 stamens arrange into 3 plus 3. In two oral, these 6 stamens are arranged in the form of 3 plus 3. And remember, this Liliaceae also, the Liliaceae has monothecus. Not monothecus. It is also has dithecus and bilobe structure. Like that, we have stamen in various families like Fabaceae, Solanaceae, and Liliaceae. Okay, remember this one also. So here, okay, so here we identify, and what we identify here. So we have to identify the characteristic feature of the family Fabaceae. So monoidalphus, no. Why? Monoidalphus condition seen in Malvaceae family. So remove it. Epiphyllus and dithecus, no. Epiphyllus are seen in, just now discussed, the stamen which is attached to the perianth. Such stamen is called epiphyllus, which is seen in Liliaceae family. So this is observed in Liliaceae, and first one observed in Malvaceae. So we have to remove these two, eliminate. Okay. Once we here see Polyadelphus, no. Generally Polyadelphus seen in, just now we discussed, Polyadelphus conditions are seen in where? Citrus. And Epipetalus, no. Why? Because Epipetalus seen in Solanaceae family. See, and here very important, Diadelphus seen in Fabaceae, and this Fabaceae has 9 plus 2, 9 plus 1. Why? Because 9 are arranged 2 bundle, it is formed, hence Diadelphus. And here Dithecus anther. So generally we have 10 stamen now. This each stamen, these 2 stamens are arranged in 2 bundles, 9 and 1. Okay, and this stamens has dithecus, means stamens are bilobed, means like this, bilobed structures, but each lobe consists of 2 theca, hence they are called dithecus. So dithecus anther are seen in Fabaceae only. So right statement, and these two, Diadelphus and dithecus, make this statement become, it is a characteristic feature of Fabaceae. So here right answer is third one. Okay, next one here, 25th question. Identify the mismatch pair. Here keyword is mismatch pair with regard to transcription in eukaryotic cell. Here giving about transcription enzymes in eukaryotic cell. So which transcription enzyme here giving? RNA polymerase enzyme. Very important. RNA polymerase enzyme is a very important trans enzymes in eukaryotic cell. It play very important in transcription process, means it helps in formation of mRNA from DNA, transcription enzymes, hence they are called transcription enzyme. Okay, fine. So here in eukaryotic cell there are 3 different types of transcription enzymes we have. How many? 3. One is RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Like that, there are 3 different types of RNA polymerase enzymes are there, and each polymerase enzyme has their specific role. For example, here RNA polymerase 1, which helps to transcribe, very important, that is ribosomal RNA, which are ribosomal RNA, and RNA polymerase 2, which helps in synthesis of messenger RNA, and RNA polymerase 3 helps in synthesis of transcribed RNA or transfer RNA. The transfer RNA means tRNA, messenger RNA means mRNA, ribosomal RNA means rRNA. Like that, there are 3 different RNA which play important specific role. And in rRNA, we have again different types of ribosomal RNA. All type of ribosomal RNA are synthesized by transcribed by RNA polymerase 1, except this one important, except 5S rRNA, very important, except this 5S rRNA, remaining all type of ribosomal RNA synthesis by RNA polymerase 1. So like 28 rRNA and also 18S rRNA, and also like 5.8 rRNA. Okay, 5.8, 28, 18, 5.8, and also we have 16S rRNA. All these type of RNA synthesis by RNA polymerase 1, except this 5S rRNA. Remember. Now coming to RNA polymerase 2, and synthesis mainly mRNA, before and precursor of mRNA is HN. So this HN RNA later, which undergoes many modification, then it become mature RNA, that is called mRNA. So means this RNA polymerase 2 helps in synthesis of hnRNA as well as mRNA. Now coming to RNA polymerase 3, which helps mainly transfer RNA, tRNA. Along with transfer RNA, it also helps transcribe the snRNA. Very important, snRNA, and also very important 5rRNA. So here 5srRNA. Here remember. So this RNA polymerase 3 synthesize or transcribe 3 different types of RNAs. One is tRNA, second one is snRNA, and rRNA. In which rRNA? 5S rRNA. Remember, 5S rRNA also transcribed by this RNA polymerase 3. Okay, right. So now we analyze here. So which one is the right answer? Just yeah. Okay. So here RNA polymerase 1, 20S, 28S rRNA, 18S rRNA. Yes, this statement is right. But here we have to identify mismatch pair. RNA polymerase 2, snRNA, hnRNA. Actually hnRNA is right, but snRNA giving. Who generally snRNA is transcribed? tRNA. So this statement is wrong generally. And polymerase RNA polymerase 2, yes, hnRNA. And this hnRNA, from this hnRNA there is a formation of mRNA. Hence hnRNA is a precursor of mRNA. So this statement is right. RNA polymerase 3, yes, snRNA and 5S RNA. See, 5S RNA. Generally, ribosomal RNA synthesis are transcribed by the polymerase 1, but except 5S ribosomal RNA synthesized by this polymerase 3. So this also right one. So here second is a mismatch pair. Why? Because in which hnRNA is there, but here again asking snRNA, but snRNA synthesis by RNA polymerase 3. So hence this statement, this word make this statement completely wrong. Which one? snRNA. If there is no snRNA, only hnRNA, also this statement is right by RNA polymerase 3. Okay, no? Right. So this one wrong. Okay, next one, 26th one. So here follicular stipules and modified terminal tendrillal leaflets are found in. So here let's start. And these questions which came from the leaf modified into tendril, leaf modified or leaf modification into tendrils. So let's start some discussion on this important concept. So generally here, if entire leaf modified into tendrils are generally seen in a plant called lathyrus plant. Lathyrus are also sweet pea plant. Except here, except stipple, the entire leaf modified into tendrils, which is seen in lathyrus or sweet pea plant. And only stipple modified into tendrils are seen in smilax. Very important, smilax. Remember, only petiole or it is a stalk of the leaf which is only modified into tendril, which is seen in clematis and nepenthes and clematis. We have seen modification of petiole into tendrils. And here one more important, and only leaf apex, get modified into tendrils, which is seen in gloriosa superba, glory lily. Very important, only that is terminal leaflet, only terminal leaflet modified into tendril, and which is seen in 𝗪𝗠 sativum. Here 𝗪𝗠 sativum, nothing but pea plant. So by this analysis, just here we have to identify which is the right one. Here leaflet, terminal tendril leaflets are seen in generally in 𝗪𝗠 sativum. Why? Because in which terminal leaflet modified into tendrils. So here smilax, where we have only in smilax only stipules modified into tendrils. Remember, in smilax stipple modified into tendrils. In glory. So continuation. Next question is 27th. Here 27th question. Hydrophytes completely submerged in water, not rooted. The mud. So here giving some examples of hydrophytes, and these hydrophytes are completely submerged. So let's discuss explanation on hydrophytes. So generally here hydrophytes are the plants which grow in water. Here hydrophytes are plants which grow in water. It's called hydrophytes. And generally there are five types of hydrophytes. That first one is free floating hydrophytes. Here free floating hydrophytes means they are freely float on the surface of water. They do not have any contact with any substrate, mud. Such plants are free floating hydrophytes. So example for free floating hydrophytes like Pistia, Lemna, and also Salvinia. So these are the few hydrophytic plants. They are free floating hydrophytes. Now second one, rooted hydrophytes with free floating leaves. Here in which rooted hydrophytes means they have root and they attach to the mud or substratum. So free floating, rooted hydrophytes with free floating leaves. But their leaves are freely float on the surface of water. Free floating leaves. So example for rooted hydrophytes with free floating leaves is Nymphea and Victoria Regia. So Victoria Regia has the largest leaf. It has the largest leaf. So in previously the largest flower is Rafflesia is the largest flower, Rafflesia, but largest leaf is Victoria Regia. It has. So it belongs to rooted hydrophytes with free floating leaves. Their leaves are freely float on the surface of water. Now here third one, submerged suspended hydrophytes. So these hydrophytes plants are completely submerged in water. They're completely submerged in water. So they do not have root. This type of hydrophytes best examples are Hydrilla utricularia. Hydrilla and utricularia. So here utricularia is also a type of insectivorous plant, and also we can use another term for utricularia, that is bladder. And one more type of hydrophytes here, fourth one, that is submerged rooted hydrophytes. Rooted hydrophytes. So this plant also completely submerged in water, but they have root. But in third one, they do not have root. They are completely submerged in water. But fourth one, submerged rooted hydrophyte, they have root, and they also completely present in the water. So best example for submerged rooted hydrophytes is Vallisneria and zoster. Vallisneria plant and zoster. Now coming to fifth one, here I am writing fifth type, and the last type is amphibious hydrophytes. So partially they are present in water and partially they are present here or towards land. So this type of amphibious hydrophytes are commonly seen in very important plant Typha and Limno. So these are the few different types of hydrophytes which is completely grow in water. Okay, so once we analyze these options here, so here asking completely submerged water, the hydrophytes which completely submerged in water but not rooted here. So third option, submerged suspended hydrophytes. So Hydrilla and utricularia. See here where we have Hydrilla and utricularia, we have to choose C. Hydrilla and utricularia, right answer. Here see, Hydrilla is submerged suspended hydrophytes, but Vallisneria is submerged rooted hydrophytes. And coming to third one, Typha and Salvinia. But Typha is a amphibious plant, but Salvinia is a free floating hydrophytes. Here Nymphia, see Nymphia, generally Nymphia where we have this one. Here 28th question, gene expression in eukaryotes could be regulated at. So generally these questions came from regulation of gene expression. So generally in eukaryotic organism, gene expression could be regulated at multi process. Generally here there are four process the gene regulation can be regulated. In that first step here, in first step, transcription level. So how transcription level the gene regulated we will discuss. Generally we have double stranded DNA like 5 prime to 3 prime to 5 prime. So from this one of the double stranded DNA act as template strand for the synthesis of transcription. There is a formation of one strand of RNA. So first form RNA is hnRNA. Here heterogeneous nucleus RNA, hnRNA will form from directly one of the strand of DNA. So this process is nothing but transcription level process. First process, this one. Okay. So after that, this formed hnRNA will undergo second steps. Second step, that is here I am writing. Second step, post level. So in post transcription, what happened in due to here this post transcription like splicing and capping process. So all this process will occur in hnRNA, then it become converted into complete mature RNA. That mature RNA nothing but mRNA. So the result of post transcriptional level, it form the mRNA from hnRNA by undergoes splicing and capping. So this whole process are occur in post transcriptional level. So the result of post transcriptional level is formation of mature mRNA, and then this mature mRNA then it undergoes third steps. So that is transport level. So in transport level, what happened here? The formed mRNA, generally where mRNA will form in nucleus only. So here nucleus, this mRNA will be formed now. So this nucleus, the formed mRNA from the nucleus, then it goes out, transport out into the cytoplasm. So here what happened in transport level? So formed mRNA, the formed mRNA transport to the cytoplasm. So this process is nothing but transport level. So here the formed mRNA will transport out into the cytoplasm. So in cytoplasm, then this mRNA will undergo translation level. So here in translation level, what happened? In transport level, the mRNA which is present in the cytoplasm. So this mRNA in cytoplasm, remember in cytoplasm will undergo translation process. Then there is a process of synthesis of protein. So in finally by the end of the translation level, there is a synthesis of protein will be synthesis. Like that, there are four important steps are regulated in gene expression. So the result of transcriptional level, formation of hnRNA. The result of post-transcriptional level, result of formation of mature RNA, mRNA. Then transport level, in which matured mRNA from nucleus into cytoplasm. In cytoplasm, it undergoes translational level, that is last level, in which mRNA undergoes translation process to form protein. So like that, there are in 4 multi-steps process the gene can be regulated. So here we have to choose. Okay, so first here we have gene expression could be regulated at, yes, transcriptional level gene can be regulated. So what is the result of transcriptional level? If gene can be regulated means there is a formation of hnRNA only, and when this hnRNA undergoes post transcriptional level, then there is a formation of mature RNA, mRNA. So here also gene can be regulated even though translational level. So what is the result of translation level? Formation of protein. Here protein will be formed from mature mRNA. And splicing process level, yes, splicing process level nothing but post level. So what happened in post transcript level? In which hnRNA completely modified into mature RNA. So here also occur. And formed mRNA transport level from nucleus to cytoplasm. This one also occurs. So here the given 4 option also right. So here we have to choose the right option here. Just wait. Yeah. So here we have it. So 1, 2, 3, 4. So in 4 level also the gene can be regulated. So next one, 29th question here. Match the following questions. So in list 1, given various different types of enzymes and their functions, means their important role. So these are questions which is framed from enzymes and its classification. Let's start. So some explanation on dehydrogenase enzyme and transferase enzyme, lyase enzyme, carbonic anhydrase. Here dehydrogenase enzyme. So this dehydrogenase enzyme which catalyze oxidation reduction reaction, very important. Dehydrogenase enzyme which catalyze oxidation and reduction reaction. Simply we can use redox reaction. Redox reaction means it undergoes oxidation and reduction both. Okay, so here in this, here the substrate 1 and substrate 2 both substrates are involved. Yes, so here substrate 1 undergoes reduced, and here substrate another substrate 2 it undergoes oxidase. So substrate become oxidized, substrate 1 become reduced. So this process A4 fourth. Now coming to transferase enzyme. So here transferase enzyme means generally this enzyme which catalyze the transfer of group. It helps in transfer of groups between pair of substrate. So there are 2 substrate we have, in which like a substrate from substrate 1 the group will be transferred to the substrate 2 here, and then it become, so here group transferred from one substrate to the another substrates. So this type of reaction is called transferase reaction. So here B is third one. Now coming to lysase enzyme. So this lysase enzyme which mainly helps to removal of group from substrate. So it main helps to remove this group from the substrate. But here in transferase enzyme, transfer of group between pairs of substrate. But in lysase enzyme, removal of group from the substrate. So if you observe here, and in which there is a here carbon X and Y here there now. So there is a removal of this group, and then it make the double bond. So here right answer is C, first one. Now coming to carbonic anhydrase. So this carbonic anhydrase which speed up the hydration process, which speed up the here, it speed up the hydration of carbon dioxide, means so carbon dioxide which combine with water, hydration, then it result to formation of carbonic acid, that is H2. So this is this reaction which catalyzed by this carbonic anhydrase enzymes. So here where we have here carbon dioxide, water, and H2CO3. So here D is second one. So like first A4 4 3 1 2. So right answer is second one. Yes. Next 30 questions 30. So arrange the correct order of chemical composition of living tissue or cell. Here we have to arrange the chemical composition of cell or tissues in terms of percentage of the total cellular mass in descending order. Here note down descending order. We have to arrange descending order. So what we have to arrange? The chemical composition of cell in terms of percentage. First, generally with a cell which contain various molecules, in which the highest, here descending order means highest to lowest. We arrange highest to lowest. For example, here in a cell, generally there are highest number of chemical composition is water. So how much water is there means generally 70 to 90% of water is there in cell, 70 to 90%. And then proteins. So protein compositions are there around 10 to 15%. And after protein, we have nucleic acid component, so around 5 to 7%. And then we have carbohydrate and CH2O or starch composition around 3%. And last, we have little amount of chemical composition in cell, that is 2% only. So like that, in a cell or tissue, we have chemical composition in this percentage. So we have to arrange all this in highest to lowest, that is descending order. For example, here how to we add means, so water is more than protein, so protein here more than nucleic acid is more than carbohydrate then lipids. So in this order we have to choose. So first water, where we have water, yeah, then protein, nucleic acid, carbohydrate, lipids. So this is the right sequence. If in questions asking descending order, ascending order, then we have to change this sequence. Like we have to take from lowest to highest chemical composition. For example, for this we have like this. So here lowest composition lipids, then lowest composition lipid. So then here lipid is less than carbohydrate, then carbohydrate is less than nucleic acid, then nucleic acid is less than protein is less than water. So like that sequence will be there if in questions asking ascending order. But in questions here descending order is given now, so we have to choose the sequence in highest to lowest. So this is the right answer. So remember, generally in many competitive exams they will ask arrange the greenhouse gases in descending order. So here arrange the greenhouse gases in descending order. So how we arrange, we will discuss. So here the greenhouse gases means descending means highest to lowest now. Yes. So the main responsible and highest gases which responsible for greenhouses, so that is what, carbon dioxide. So how much means carbon dioxide, generally 60% of carbon dioxide is responsible for green gases. Then methane gas CH3, then CH4, how much means 20%. And then chlorofluorocarbon CFC, how much means 14%. And then last nitrogen oxide, how much means 6%. So these are the value which is responsible for greenhouse. So here we have to arrange them descending order means first highest concentration carbon dioxide 60, methane 20%, chlorofluorocarbon 14%, nitrogen oxide 6%. Like that we have to arrange in descending. If in asking in ascending order of greenhouse gases, then we have to make this sequence like this. How means, so lowest what we have, nitrogen oxide is less than chlorofluorocarbon and is less than methane is less than carbon dioxide. So this is the order of ascending of greenhouse gases, but here this is the order of descending order of greenhouse gases. So remember this. So here 31st question, here 3.1, which of the following restriction sites are present in ampicillin gene of plasmid PBR322? So here vector, so here artificially a restructured vector here given, that is PBR2 vector. So it is the most common vector used to gene transfer. So if you see the structure of PBR322, it has vectors are plasmids are double stranded, extra chromosomal single stranded DNA. So in which this PBR322 has 4 important yet unique characters. We know that already we discussed in many times. So they are Ori sequence, origin of replication sequence, and it also has low molecular weight. And the third, it has selectable marker. It has selectable markers. Generally we have 2 selectable markers in which, and here okay, so selectable marker we have 2. So here right side, left side, here right side we have ampicillin resistant gene marker, and here this side left side we have tetracycline resistant gene. Okay. And one more important characteristic feature of this PBR322, it has many cloning sites, cloning or restriction sites. So generally in PBR322 we have 8 restriction sites. Okay, right. So here in questions, so which of the following restriction sites are present in ampicillin resistant gene of plasmid? So remember, in ampicillin resistant gene, there are two cloning restriction sites are present here. So they are PST and here PVU1. So this PST1 and PVU1 are the restriction sites which is present in the ampicillin resistant gene. So likewise, in tetracycline resistant gene, we have two restriction sites. So they are BAM and also SAL. So like that, there are four restriction sites are present on both selectable marker like ampicillin resistant and tetracycline resistance. Okay, and along with these four, and there are also again more four restriction sites. Total there are 8 restriction sites are present now. So more 4 restriction sites also present. So here in here we have one restriction site, that is PVU2, and here in between these three we have 3 important restriction site. For example, here we have 𝗖𝗎𝗏 restriction site, and then here we have 𝗪1, and here we have HIND. So like that we have 3 different types of restriction site in between ampicillin and tetracycline resistant genes. Okay, like that here total there are 8 restriction sites are present, and PVU present in a rope gene. So replication of protein gene here we have, and here we have sequence called ORI sequence. Okay, so likewise we have 8 restriction site like 𝗖𝗎𝗏1 and 𝗪1, HIND3, BAMH1, SAL1, PVU2, and PVU1, PST1. So all these are 8 restriction sites. But here in questions, they are asking which of the following restriction sites is present on ampicillin resistant genes of plasmid, means that is PST1, PVU1. We have to choose these two. Where we have here PVU1 and PST1. So A and B is the right answer. Okay. If in questions they are asking means like this, tetracycline. So identify which of the following restriction sites are present in tetracycline resistant gene of plasmid PBR, means then BAMH1 and SAL1. These two are tetra resistant gene. So but in here we have to identify ampicillin resistant gene of plasmid, that is here. Sorry. PVU1, PST1. Okay. So next one, 32. So this question which is from biomolecules, especially in carbohydrate. Bio in that carbohydrate biomolecules. Here question here, inulin is a polymer of, not insulin is completely different. So here insulin is a hormone, proteinous hormone. Remember, it is secreted from beta cells of pancreas. Okay. Due to deficiency of insulin, it leads to a disease called diabetes mellitus. But here asking inulin. Here inulin. So inulin is a polymer of what? Glucose, fructose, galactose, and raffinose. Generally, inulin is a naturally occurring. Here I am writing. Inulin is a naturally occurring storage this is a storage polysaccharide, and which is found in many plants, like especially in chicory plant, and it is a storage polysaccharide. Remember, it is a storage polysaccharide. And this insulin, which is made by the polymer of fructose, very important. It is a polymer of fructose. So due to polymer of fructose, which made the polysaccharide, that is inulin, and it is a naturally occur storage polysaccharide. So right answer here, fructose only. So next one here, 33 question. Match the following type of question. Here list one, giving elements, their physiological role and physiological disease deficiency. Okay. First about manganese. Here we need to discuss some importance of manganese here. Yeah. So manganese is an activator of, very important, it is an activator of indolactic acid oxidase enzyme, and its toxicity leads to cause a disease called brown spot disease. So here we have to identify manganese. Here it is an activator of indolactic oxidase, and this cause brown spot surrounded by chlorotic veins. So here this pair is wrong. Okay, it is not helps in anion cationic balance. Okay, but disease is right, but this physiological role is wrong, hence this statement is completely wrong. Now coming to zinc. Generally zinc is a oxygen synthesis. Here we have to choose needed for oxygen synthesis, means 2 is C. Here this one, list 2. But due to deficiency of zinc is leads to cause molted leaf disease. Okay, and here this role is completely wrong. Okay, and now coming to boron. Here boron is a calcium uptake and utilization, which helps in calcium uptake and utilization, this one. And translocation of food and pollen grain germination, all are the various function of boron. The deficiency of this disease which leads to heart rot disease. So here R. And chlorine, finally here chlorine we have. So this chlorine helps in anion and cationic balance, here A. Okay, and the disease which is caused in legume, that is bronzing disease, means yes. So we have to choose, okay, which is the right one answer here. So we have to choose. One is D and P. One, yes, manganese. One is D and P. Just wait here. Manganese. Here D is what? Activator of indole acid oxidase. So D, and here P. Brown spot surrounded by chlorotic veins. Coming to zinc. So zinc is a needed for oxygen synthesis. Hence 2C, and deficiency of this cause motor lip disease. Hence Q. Boron helps in uptake of calcium utilization. So here B, and the resulted structure cause a disease called hot rod disease, means R, BR. So here third one is BR. Where we have BR? So here third one BR, here we have. Now coming to chlorine. So chlorine is helps in anion and cation balance, means A, and result to form bronzing in legumes, yes, AS. Here we have. So like that we have to choose. The right answer is fourth one. Next 34 question here. So which of the following genes were introduced in cotton to protect it from cotton bollworms? So these questions which is from the Bt toxin application in biotechnology. So generally what Bt toxin will discuss. Bt toxin. So Bt toxin protein. Generally this Bt toxin protein produced from a soil bacteria called Bacillus thuringiensis. So it is a soil bacteria. So this Bt toxin, which is encoded by a gene called Cry gene. Very important. Cry gene. So this Cry gene which encode this Bt toxin protein in Bacillus thuringiensis. Okay. There are two different varieties of Cry genes are present, like in which we have Cry1, and also in one series Cry, in two series Cry2 AB. Generally here see this first Cry1 AC and Cry2 AB. These two genes which mainly helps to control cotton bollworm. These two control cotton bollworms. But this Cry which control car-borer disease in cotton. Okay. So but here in questions, which of the following genes is introduced in cotton to protect from cotton bollworm? So which disease, which gene? Cry1 AC, Cry2 AB. Cry1 AC and Cry2 AB. If it is only asking about corn borer, then only one gene which responsible for this, so that is Cry1. So but here answer is fourth one. Why? Because this Cry1 AC and Cry2 AB which responsible or which protect from the cotton bollworm. So next one, 35th 35 questions. Function of plasma membrane extension called mesosome in prokaryotic cell. Here we know that prokaryotic cells are mostly bacteria, in which the outermost is cell wall, and here we have some extension of plasma membrane like this. This is what plasma membrane or cell membrane. So this extension of plasma membrane is known to be as a mesosome. So these mesosomes in prokaryotic cell perform various functions, like mainly which helps in cell wall formation, and which also helps in DNA replication, and also which helps in respiration, and also helps in secretion process. So these are the important functions which is performed by this mesosomes, and mesosome is the extension of plasma membrane in prokaryotic cell. So here all are the functions which is performed by this mesosomes, hence right answer is third one. So next 36th question here, assertion reason type of questions. So here assertion, E. coli having PBR322 with DNA insert at BAM-H1 site cannot grow in medium containing tetracycline. So recognition site for BAM-H1 is present in tetracycline region of PBR322. So we have to identify correct statement. So let's discuss. Just now we discuss. So here it is what PBR322 classmate, PBR322. Here we have two selectable marker. So here right side we have ampicillin resistant gene, and here left side we have tetracycline resistant gene marker. So here this ampicillin resistant gene has two important site. They are PVU1, and also it has one more important structure, that is PST1. But here it has BAM1 site and also SAL1 site. So like that we have four recognition or restriction site on this two selectable marker. Okay, fine. So here in these questions, if we insert DNA, foreign DNA, if we insert foreign DNA or desired DNA or gene of interest, we can use any term, desired DNA or foreign DNA or gene of interest, where at the BAM-H1 site here. So if you introduce here, Just wait. If you introduce this foreign DNA here, this foreign DNA or desired DNA into this BAM-H1 site here. If you introduce here at BAM-H1 site and cannot grow in medium containing tetracycline, means yes. Why? Because so it is disturb this genes which can disturb this tetracycline containing medium. Yes. So which mainly, so when we insert foreign gene at BAM-H1 site of PBR322 which disturb the, here I am writing, when we insert foreign DNA at BAM-H1 and which disturb the which gene, tetracycline resistant genes. So when it disturb the tetracycline resistant genes and know this E. coli cells from growing on medium containing, which prevent the E. coli cells from tetra media. So this complete process is said to be as a insertional inactivation method. So here insertional inactivation means in which we insert a foreign DNA at the site of tetracycline, especially in BAM-H1, then and these E. coli cells cannot grow in tetracycline culturing media. So here both, if you read these two options clearly here, see, so E. coli having PBR3 with DNA inserted BAM-H1 cannot grow, yes, so cannot grow medium containing tetracycline, means assertion question is right, correct statement. This one correct statement. Recognized site for BAM-H1 is present in tetracycline region of PB. Just now we discussed. Yes, where we have BAM-H1 site at tetracycline resistant gene. So hence which prevent the E. coli cell from growing tetracycline containing medium. So this type of inserting of foreign DNA to prevent the E. coli cells from their respective gene antibiotic resistant genes, such processes are called insertional inactivation method. Okay, next one, 37. Which of the following is correct answer regarding the structure of cilia and flagella? Here these questions came from cell biology. In cell biology, which is asked from cytoskeleton. In cytoskeleton, asked about two important structures called cilia and flagella. The cilia and flagellum also appendicular structure. They help for attachment and movement. Flagella. Okay. So here, so before we analyze these options, let's start what exactly the cytoskeleton and what is the structure of internal structure of cilia and flagella we will discuss here. So here generally cytoskeleton here. Cytoskeletons are proteinous filamentous. These are proteinous filamentous network-like structures present in cytoplasm of the cell. Generally, the cytoskeletons, which is mainly made up of three important components. So they are most important component in cytoskeleton, that is microtubule, and second one is micro, and third one is interfilaments. So these three components together form cytoskeleton. So generally, these microtubule are seen in the arrangement of microtubules is like 9+0 arrangement, or they are also present in this 9+2 arrangement. Here this 9+0 arrangement of microtubules are generally seen in two structures. One is centrosome, means centrioles, and also which is seen in basal bodies. So here basal bodies are centriole is a one type of cell organelles which is present only in animal cell, which is absent in plant cell, remember. And these basal bodies which is generally present and which helps to anchor the base of cilia and flagella. So at the base of cilia and flagella structure, we have at their base of cilia and flagella we have the anchor structure that is called basal bodies. And this centrioles and basal body shows 9+2 arrangement of microtubules. Now coming to 9+2 arrangement. So here these 9+2 arrangements are generally seen in cilia and flagellum. In these two structure we have seen this 9+2 arrangement. So how they are arranged we will discuss. First we will discuss 9+2 arrangement. So here in this 9+2 arrangement we have different types of different structures, like so at peripheral we have doublet of microtubules. For example, like so here, so this is one doublet, two microtubules together form like this peripheral. 3, 4, 5, 6, 7, 8, 9. So like 9. See what is this? Peripheral microtubules. So they are present in doublet, two doublet. Okay. So 9 pairs which is present at the edges of cilia or flagellum, and their pair means doublet. Okay. And remember here in center we have two singlet. They are separate singlet. So here I am writing central microtubule. Here we have central microtubules. So but the central microtubules are singlet, means they are single, not pair. But peripheral microtubules are doublet. Here pair, but here single. Okay, the single is called singlet. Here pair means doublet. And here these central microtubules are covered with a sheath, which is covered with a sheath like this. Now this sheath is called central sheath. So here the central sheath is only one, and which is a protective sheath around the central tube microtubules. It is a protective sheath around central microtubules which is present. And here very important structures. So that is this doublet peripheral microtubules, doublet which connected with singlet central microtubule with the help of radial spoke like this: 1, 2, 3, 4, 5, 6, 7, 8, 9. So these 9 are what? Radial spoke. So how many? 9. So there are 9 radial spokes which connect peripheral microtubules with central micro. So these are the structure of micro in 9+2 arrangement. Okay, no? Right. So what is the importance of radial spoke? Which mainly helps to connect peripheral microtubules to the central microtubules. But remember, central microtubules always in singlet, but peripheral microtubules always in doublet. So in some exam they will ask the total number of microtubules in cilia or flagellum. If they ask like that, then we have to count. So in peripheral we have how many? 9. So 9 always pair. 9 to the 18 pairs, 18 microtubules. But at center we have 2 plus 2. Then 20 total 20 microtubules are present in cilia and flagellum. Okay, and then here now we move on 9 plus 0 arrangement. How this 9 plus 0 arrangement will be having, we will discuss now. Okay. Next here see 9 plus 0 arrangement of microtubule seen in two important organelles. They are centrioles and basal bodies. Yeah, how they are arranged we will discuss now here. So in peripheral, like 9 plus 2 we have doublet now, but here we have triplet. In peripheral we have 9 triplet structures at peripheral. So here like this 9. So one triplet 1 2 3 like A B C, but here we have only A and B doublet. One more A B C like this. There are 9. See 1 2 3 4 5 6 7 8. Here just we will adjust 9. So there are 9. See, there are 9 peripheral microtubules in triplet. Very important, triplet. In 9 plus 0 we have triplet peripheral microtubules. So here at center, very important, at center we don't have central tube. Central microtubule is absent. Very important. At central microtubule is absent. Here we don't have micro. Instead of central microtubule, instead of which we have a protein rich core structures. Protein rich core structure. That protein rich core structure is called hub. Very important. So here like, so instead of central microtubules we have protein rich here, protein rich structures like this. So this structure is called hub. Okay, so central microtubule is absent, very absent. So instead of central microtubule we have protein rich core structure called hub. So this structure is called hub. Okay. So then these peripheral microtubules are triplet in nature. They are connecting to peripheral to this hub with the help of radial spoke here like this. So there are how many radial spokes are we have? 9. So here also we have 9 radial spoke. Here 9 radial spoke is there, and this radial spoke helps in connecting peripheral microtubules with hub. Why? Because here at center we don't have any microtubules. So hence this peripheral microtubules, they are in triplet in nature. They are connected to the hub with the help of these 9 radial spokes. So these are how the microtubules are arranged in centriole and basal bodies. This one. And here we discuss how these microtubules are arranged in cilia and flagella. So by analyzing all these things, now we will analyze these options like here. Just wait. Yeah. So first read questions carefully, then we move on the options. Okay. Yeah. See here. Okay. Fine. Read first. Here giving peripheral microtubules doublets. Okay. And read first question. So choose the correct answer. regarding the structure of cilia and flagella. So generally just now we discussed cilia and flagella, which type of arrangement of microtubules are there, 9 plus 2, remember. Okay, right. So here peripheral microtubules, they are doublet, central microtubules are singlet, just now we discussed, and radial spokes and central sheath. So central sheath present around the central microtubules as a protective sheath. Okay, right. So 9 plus 2 here, peripheral microtubules, 9 plus 0, no, wrong. Okay, 9 plus 0 is wrong, and 9 plus 0 is a type of arrangement of microtubules which is seen in centrioles and basal bodies. But here we have to find out peripheral microtubules must and should be doublet. Here, see, 9 plus 2 arrangement which is seen in flagella and cilia. But we don't have how many number of peripheral microtubules doublet here, not mentioned. Just it is mentioned this type of arrangement are seen in flagella and cilia. Okay, this one also made wrong. See here, how many peripheral microtubules are there? 9. So these 9 represent what? Peripheral microtubules. These peripheral microtubules present in doublet. So this one right. And central microtubules, how many are there? 2. Yes, just now we discussed now. Here central we have 2. So at peripheral we have 9, like this. Okay, like this. So 9 we have now. Right. At center we have 2. This one right. How many radial spokes are there? 9. Why? Because each radial spokes which connect the peripheral doublet microtubules with central singlet. So this one right. How many central sheath we have? 1. See, this central sheath which is present, the central sheath which is present around these central microtubules, like that. So which one is the right answer here? C is the right answer. Yes, here giving in official key also right answer is C. If in some time, if in exam they will ask total number of microtubules in central or basal body, if they ask these questions, then your answer must be here. How many microtubules are there at peripheral? 9. But 9 are present in which form? Triplet, means 9 3s are 21. So 21 microtubules are present in cilia and basal bodies. But in 9 plus 2 arrangement, how many? 9 2s are 18. Why? Because 9 in doublet, but at center we have only 2. So 2 here. So 18 plus 2, 20. So 20 microtubules, total microtubules are present in flagella and cilia. So but 21 microtubules present in centriole and basal body. Remember this, very important. Okay, next one here, choose the incorrect pair. Here we have to choose incorrect pair only, remember. So here giving microbes, their product, their application. See here, first MCQ here, streptococcus, from which we obtain streptokinase, yes, and which helps in clot removal from blood vessels. Yes, this statement is also right. Okay, and why? Because this streptococcus is genetically modified bacteria from which we obtain this streptokinase, which helps to remove blood clot from the blood vessels. Okay, fine. And Clostridium botulinum, its product is lipase. No, wrong. Clostridium botulinum product is butyric acid. Butyric acids. But their function is same. Lipase function is oil stains removal. Okay. Actually, lipase function is same, right? Lipase function is to remove oil. But here, the product of Clostridium botulinum not lipase, it is a butyric acid. So this statement is wrong. Coming to Monoscus purpureus. So it is a fungus. So in fungi, it belongs to yeast. It produces statins. Yes. So the important application of the statin is lowering the blood cholesterol. And coming to Trichoderma palisporum, it is also belongs to fungi, and it produces cyclosporin A, and it is act as a immunosuppressive drug. Yes. So here the second one is the wrong one. We have to choose incorrect pair only. Why? Because why second one is wrong means here Clostridium botulinum produce a product called butyric acid, not lipase. Okay, right. So here 39 questions. BOD of wastewater, sewage water, is estimated by measuring the amount of here. So this question from sewage treatment, biology in human welfare. So here BOD means biochemical oxygen demand. Here biochemical oxygen demand. So highlight this oxygen. Or also we can use another term, biological oxygen demand. So what is it? BOD. So generally BOD is estimated by measuring how much amount of oxygen is consumed. Important here, which is measured by oxygen consumption by micro in wastewater or sewage water. So here this BOD value we can measure by how much amount of the rate of oxygen consumption occur by microorganism in wastewater or sewage water, we can call BOD, biological oxygen demand. Okay, and this BOD value is directly proportional to the oxygen consumptions. For example, means, and here high BOD value is there, means high BOD value is directly proportional to the amount of biodegradable, the amount of bioorganic matter present in waste. It means that if, for example, in a wastewater, sewage water, if the BOD value is high, means the water is highly polluted. Why? Because this BOD value is directly proportional to the amount of biodegradable organic matter present in the wastewater. Okay, so wastewater. So hence, if BOD value is more, then higher pollutant. If BOD value is less, then less pollutant. Remember. So when we want to measure the BOD value, there must and should be the oxygen consumption occur by microorganism to degrade this organic waste material in sewage water. Okay, so remember, and in which what happened? So here, so oxygen evolution, no. Here, by oxygen consumption, no. Here, biodegradable organism. So very important here, need to remember. So BOD of wastewater is estimated by measuring the amount of biodegradable organic matter. So right answer is biodegradable organic matter. Okay, but here we have to estimate the oxygen consumption. Here oxygen, how much amount of oxygen consumption occur, but the right answer is here this this microorganism use this oxygen has a demand by microorganism to break down this biodegradable organic matter, to break down this biodegradable organic matter. So there is a more need of oxygen. If more need of oxygen is occurring, means there is a more biodegradable organic matter will be undergoes degradation process. Okay, remember. So last MCQs in botany paper analysis, that is for 40. Here choose the correct match in relation to substance and their function. Here giving substances, here 4 substances are giving and their functions are giving. We have to choose correct pair only. Okay, right. GLUT4. Here GLUT4 means glucose transport 4. So here this glucose transport means which enable the cells to synthesize glucose. Enable the cell to synthesize glucose. Yes, this statement is right. Why? Because this GLUT4 which helps to transport glucose into fat cell and muscle cell. In fat cell and muscle cell, this GLUT4 protein which helps to synthesize or which enable to cell to synthesize this glucose in fat cell and muscle cell. For that, there is a requirement of GLUT4. So right statement, correct statement. Coming to collagen is excretory ground subs. No, collagen is not a excretory product. Okay, why? Because this collagen is the most abundant protein in the world. Very important. Collagen is the most abundant protein present in the world, and which is mainly present intercellular ground tissues. So generally this collagen are present in intercellular means matrix, intercellular or matrix of tissues, where we have seen this collagen is mostly abundant in the world, but it is generally present in intercellular tissues. So this statement is wrong. Coming to C, insulin is an enzyme. No, insulin is not a enzyme. Insulin is a hormone. And this hormone, insulin hormone, produced from beta cells of pancreatic islets of Langerhans. Islet of Langer. Okay, but hormones are mostly proteins. Remember, hormones are mostly protein, but insulin not a enzymes. Remember. So this statement is also wrong. Coming to here, so antibodies fight infection agent. Yes, this statement is right. Why? Because here, in first A statement, GLUT4 which enables cell to synthesize glucose. No, not synthesize the glucose. They only helps to transport glucose into the cell. So remember. So here I made a mistake. So that is GLUT4 which not helps to synthesize glucose in fat and liver cell, fat or muscle cell. Only which helps to transport, but only it helps to transport glucose into fat cell and muscle cell. But here in question, synthesize is giving. So not synthesize, only transport. So hence this statement is also wrong. Clear? So here the right statement is antibody is produced from lymphocytes of white blood cells and our from immune system, and this helps to fight infection against antigens or foreign pathogens. Okay na? So in this the fourth option is right one. So this is all about the agriculture and pharmacy EAPCET on 5th May 2026 shift 1 paper, botany paper analysis. Then we move on zoology paper analysis. We will also discuss zoology paper analysis like this. Okay, so thank you, have a nice day.